Good morning and welcome to the Centers of Excellence at the University of Utah Iron and Heme Center virtual seminar series. We're so excited to see all of you joining us today, and we're very pleased to have Dr. Angelo de Alessandro speak to us. Just a couple of housekeeping things for those of you who are in attendance today, attendance virtually. We will ask you to mute yourself so that we don't have that background noise going on in there. And if you don't, I'll mute you so that we can make sure that the talk goes smoothly. Dr. De Alessandro has agreed to have questions during the talk. And so if you have a question, raise your hand. I'll try and monitor things. You can post them in the chat if you want. And I will try and invite you in to ask a question during the talk, or you can hold it till the end. Either way is fine. Let me think. Anything else I want to say? I think that's it. This is being recorded and it will be posted on the CIHD's YouTube channel. It gets posted usually a day or two after, in case you're interested in reviewing what's been said, or you want to tell some friends what a great talk you just heard. And with that, I will turn the time over to our center leader, Anna Bowden, for the introduction.
Thanks, Diane. Okay, it is my distinct pleasure to introduce Dr. Angelo D'Alessandro, who is a tenured full professor in the Department of Biochemistry and Molecular Genetics at UC Denver Anschutz School of Medicine. He's the founder and director of a fantastic metabolomics core there and the director of the mass spectrometry shared resource. At age 40, Angelo has published over 620 papers, and his research is sponsored by NHLBI, NIDDK, NIGMS, and BARDA. He's the founder and CSO of Omics Technologies and serves as an advisory board member for Humanex and Mako Pharma. He's an AABB Hall of Fame inductee, Botcher investigator, and Jean Juilliard Innovation and RISE awardee. He's also an affiliate investigator to Vitalant Research Institute, the Linda Cernick Institute for Down Syndrome, the Gates Grubstake Center for Regenerative Medicine, and the Trauma Research Center, and also a wonderful collaborator. So I'm excited to hear about his work today. Thank you, Angelo. Please take the floor.
First of all, thank you so much for the kind invitation. Let me hide the floating controls. You should be able to see my screen now.
We can.
Perfect. Fantastic. Thanks so much for the kind invitation. I was making jokes about this being recorded, and I haven't fixed my hair today. But anyway, please feel free to interrupt me anytime. Unfortunately, I just hit the floating bar, so I cannot really see chats and comments. But otherwise, please feel free to stop me anytime if any question arises. Okay, finding disclosure first. Since I'm going to have a lot to present, I'm going to fly through this slide and I'm going to highlight the most important disclosure I usually present at the beginning of any presentation. During this today's talk, I will say multiple times, we did this, we did that. When I say we, it's actually the people on this slide I have the pleasure and honor to work with on a daily basis. They are from associate research professor all the way to undergrads in the lab. We generate the data through our mass spectrometry-based approaches. And it's a pleasure and honor to be working with them daily. So, as Dr. Boden mentioned, I come from the Department of Biochemistry and Molecular Genetics at CU Anschutz. And for those of you who are not familiar with our environment, we're standing on the shoulders of some giants there. Thinking, for example, Dr. Tomczak, who is famous and a Nobel laureate for his work on ribozymes. And as such, we have a strong influence, a strong commitment of the department in the space of research in the field of RNA. So I spend usually my first 10 minutes of any presentation I give, try to convince people that it's a worthwhile endeavor to study something else that doesn't necessarily rely much on RNA, and I'll show you why in the next few slides. Mature blood cells, and I try to do so by providing numbers. The first of which is something that is probably going to resonate with this audience. Red blood cells are by large and far the most numerous cell in the human body. Approximately 83% of the total cell in an adult individual, 25 trillions out of the estimate 30 trillion in an adult individual, are red blood cells. And I'm somebody who studies metabolism, so with an average lifespan of 120 days in circulation, it means that red blood cells, from a metabolic standpoint, from a thermodynamic standpoint, offer a unique challenge in that we are daily using a lot of energy to generate de novo zero point 0.2 trillion red blood cells, and while getting rid of a similar amount of those red blood cells and recycling the building blocks, small molecule metabolites that comes from the digestion of the macromolecules of these red blood cells, including iron. From a metabolic standpoint, red blood cells also offer a unique model in that during maturation you lose nuclei and organelles, so they're devoid of the novel protein synthesis capacity, and as such, they respond to environmental stimuli, including hypoxia and oxidant stress, through a mechanism of metabolic reprogramming or metabolomics-based regulation of post-translational modifications, such as, for example, phosphorylation. As red blood cells progressively, red cell precursor progressively mature from proerythroblasts all the way to reticulocytes and mature erythrocytes that lose nuclei and organelles to make room for hemoglobin. And with up to 250 to 270 million molecules of copies of hemoglobin per mature red blood cells, and heme and iron coordinating oxygen in each one of these subunits, that means that full oxygen saturation, you have anywhere between, and estimates there vary, but up to 1 billion molecules of oxygen per mature red blood cells. And since all of this, as I mentioned, is coordinated by iron in the prosthetic group heme of hemoglobin, that also means that 66% of bodily iron, 2/3 of bodily iron, is hosted in mature red blood cells, 2.6 grams, I think it is. So that also means, not just from catabolic or anabolic standpoint, if you want just to focus on metabolism, it also means that red blood cells offer a unique system to investigate responses, metabolic responses to oxidant stress. So metabolic reprogramming response to reactive oxygen species formation through Fenton-Abreu chemistry. All of this in, again, the absence of de novo protein synthesis because of the lack of nuclei and organelle. I tried to convince you in the previous slide that red blood cells are a simplified model of eukaryotic cell. We and many others are actually challenging the concept that red blood cells are just hapless sac of hemoglobin. Indeed, we now know that red blood cells actually host several thousand proteins based on most recent proteomics estimate. It has been argued that the sensitivity to the latest generation of mass spectrometers allows for some degree of contamination in these estimates with proteins derived from either plasma or other circulating blood cells or reticulocytes that impact those estimates. Actually, to address this question, we recently collaborated with Dr. Crystal Hudson at Columbia, an immunologist with expertise on blood cell biology. And we literally spent like over 2 weeks of flow sorting time to obtain the purest red blood cell population to date. So at first, we collected blood Buffyco depleted from regular blood donors. I think this was, we did both from 5 and then repeated on 10 blood donors. And then we leukocyte filter with a specific filter to remove log4, so 99.99% white blood cells, and log 2.5 platelets. And ultimately, we then flow sorted to remove residual white blood cells, residual platelets, and residual reticulocytes, and flow sorted to positively enrich for CD35 positive, CD71 negative mature erythrocytes. And on this extremely pure red blood cell population, we ended up performing all sorts of combined proteomics analysis with the SDS page combined with the gel band excision and long gradient proteomics, and then 2D reversible phase strong cation exchanges, reversible phase chromatography, and a series of other methods that I will spare you today. But ultimately, once we do all this analysis, we identify over 3,775 unique proteins in the mature erythrocytes, including coverage of hundreds of transporters and receptors that have been previously estimated or suspected to be present in red blood cells, or identified through Western blot-based approaches or fluorescent-based approaches, but never identified before through proteomics estimate. Ultimately, our compiled data set adds up to over approximately 1,000 proteins, give or take, more than all the previous proteomic studies combined in the literature. But the fact that we also have the ultra-purified population, it also shows that approximately 1,000 proteins that have already been reported in some of these proteomic studies without purification of the red cell specific subpopulation were indeed including some contaminants in the red blood cell fraction. All of this is going to be made available in what we call the deep red. I don't know if any of you is a fan of the 1980s Italian thriller movies. It's inspired by Dario Argento's masterpiece. But all of this, is going to be interactive portal that will be amenable to interrogation and linkage to UniProt databases. I will spare you that we also have mapped all post-translational modifications in this deep red database that is also amenable to interrogation. And one thing that I'm really proud of, we are not only looking at the red blood cell proteins from a qualitative and quantitative standpoint with the spiked-in stabilized off-label Q-concats, quantitative concatamers, isobaric peptides that we spike in a known concentration and we use to identify absolute concentration of given proteins of interest. But also, we can use chemical cross-linking agents such as the succinyl superate or the NTMM to introduce covalent bonds, chemical cross-link in between epsilon amine groups of lysine and carboxylic group of aspartates and glutamates of within proteins or amongst proteins, so in between intermolecular or intramolecular covalent bonds for residues of the aforementioned amino acids that face each other within up to 20 angstrom in the zero-length cross-linker approach. Ultimately, we use all this information to map the interactome at the membrane level with an interactome orbiting around band 3, cytoskeleton interactome around tyrosine, and a cytosolic interactome around anything that bounces, stumbles across upon hemoglobin alpha and beta chains. We can use all this information, especially the qualitative and quantitative information, including estimated copy number per cell, to inform reconstruction of the red blood cell metabolome that are informed by genomics data and proteomics information. We did this recently with Dr. Bernard Paulsen. The 1.0 version of the RBC gem has just been published in the PLOS Computational Biology a month ago, and we are, along with Deep Red, will be publishing the follow-up expansion to this reconstruction. And ultimately, what you can do with this, you can simulate all the reactions. At the very least, you can borrow known KM for all enzymes and superimpose the protein concentration, the protein abundances in a given sample of interest, and then simulate how metabolism would respond to perturbation, including stimulation of specific receptors, inhibition of specific transporters, pharmacological or genetic ablation of specific metabolic enzymes. If I convinced you that red blood cells are more than a helpless sack of hemoglobin, we are now trying to use leverage this proteomics information to determine whether red blood cells can perform activities that have been historically only metabolic activities in particular that have been historically attributed to specific organs. For example, transaminase activity in the liver rather than, for example, creatine metabolism or branched keto acid amino acid metabolism, detoxification of metabolites of exposure, a point I will touch upon in the last few slides of this very presentation today. Ultimately, we proposed kind of, a provocative concept that red blood cells may function as a sort of a circulating organ, 2.5 kilos in weight, that touches upon all the other tissues and organs while circulating through the body, 1 minute per circulatory transit, up to 174,000 circulatory transits in a lifespan, ultimately impacting the homeostasis either by subtracting or offering specific small molecules as a crosstalk between the red blood cell organ and other tissues. How do we test all of that? Well, first, It is not a novel concept that looking at small molecule in circulation represents a treasure trove of information for as a window on systems metabolic health. We have been doing this now for almost a century in the context of inborn errors on metabolism and the early days of clinical chemistry. We still measure transaminase activity, lactate, glycemia, and so on and so forth in blood. We want to expand that by implementing metabolomics approaches. And this is the only technical slide, I promise. We're using mass spectrometry-based tools to look at small molecules in a mature blood cell, specifically anything below 2 kilodalton. And when you pop red blood cells, lyse red blood cells with specific combination of organic solvent and aqueous phase, you will see that the majority of the spectrum is dominated by approximately 50 to 60 peaks. There are the most abundant metabolites. However, you see here in the so-called graphing jargon, these little dots that you can actually zoom in because of the sensitivity of the instrument, and you can assign a name to based on chemical physical property, including accurate tinted mass, isotopic pattern, retention time, elution through the chromatographic column, and a series of other parameters. You can do that for small molecule metabolites, hydrophilic metabolites, but also hydrophobic metabolites in a gradient of obesity, including the whole lipidome compartment. And one specific characteristic of our lab is that we develop ultra-high throughput method to afford the analysis of metabolomics, performing metabolomics analysis of any given matrix of interest, red blood cells, but any other cell, and also including biofluid from very limited matrix availability, for example, as few as several thousand, 2,000 is the limited, the least we've done, hematopoietic stem cells from flow sorted subpopulation all the way down to 0.5 microliters of cerebrospinal fluid or rare biofluids. If you're interested in reading more about this, I added a whole thematic book for Springer Nature on high throughput metabolomics, and we are coming out with the next edition as soon as all the contributions are finalized. Don't take any royalties for this, and nothing to disclose there. So if I convince you that red blood cells are important per se as a biological question in a simplified model of eukaryotic cell, if you may, it is also true that red blood cells are an extremely important drug. They're actually the most commonly issued drug, prescribed drug in hospitals in the United States, around the world every year after vaccination. Approximately 100 million units of packed red blood cell are transfused every year to all sorts of categories of recipients, both chronically transfused recipients like patients with sickle cell disease or hemoglobinopathies requiring transfusion, or patients requiring massive transfusion, for example, after trauma and hemorrhagic shock, the leading cause of mortality under the age of 59, the largest global cause for total life years lost. To the point that packed red blood cell transfusion has been estimated to be the single human invention that saved the most lives in the whole history of mankind, along with antibiotics, synthetic fertilizers, and pasteurization. Approximately 1 billion estimated lives saved. Unfortunately, unlike fine wine, as packed red blood cells, as red blood cells are stored in a blood bag for up to 42 days in common storage additive solution, they progressively lose the viscosity phenotype to acquire an echinocytic and ultimately spheroechinocytic or spherocytic phenotype that make these red blood cells that are ultimately less deformable, more likely to be sequestered in the splenic slits and erythrophagocytosed, undergo extravascular hemolysis. Only a limited amount of red blood cells, less than 1% per Food and Drug Administration regulation, or less than 0.75 if you look at the Canadian or European Council guidelines, actually hemolyze in the blood bag, are likely to hemolyze upon transfusion in circulation, intravascular hemolysis. Most of the red blood cells that get removed from the bloodstream after transfusion, they don't just pop, they're actually sequestered in the spleen first and the liver next. We can actually determine the quality of transfused product by radiolabeling with chromium 51 prior to transfusion of a small amount, usually 10 ml, of packed red blood cells into autologous recipients. So when trying to license new product, the donors also become the recipient of their own blood at the end of storage, and they just receive a fraction of 10 ml of the radiolabeled red blood cells, stored red blood cells that they themselves donated. And ultimately, we can look at the distribution across 2 decades of post-transfusion recovery studies, several hundreds of studies, several hundreds of chromium-51-based radiolabeling survival studies. Dumont and Buchon showed that indeed there is a wide distribution of post-transfusion recovery across the donors in multiple studies. In particular, some donors store really well above the 85% threshold. For Food and Drug Administration, the European Council guideline, the minimum threshold is 75%. But then some donors actually, approximately 20 to 30% of the donors do not meet this threshold, suggesting that there are factors that impact storage biology that are at least somewhat donor-related. So in an attempt to investigate what are the factors that contribute to the heterogeneity in the quality of stored blood product and ultimately to the extravascular hemolysis of stored and transfused packed red blood cells that limit the efficacy but also potentially increase the potential side effects of transfusions, such as acute respiratory distress syndrome or septic complication. And I will not go much into details of that today. We collaborated with a team of investigators, literally a whole village of investigators across multiple institutes, including Vitalent Research Institute as part of the REDS RBComics study, and collaborators at the Jackson Laboratory, UCSF, RTI International, UCSD, INSERM in Paris, and here at CU Anschutz. And ultimately combine all of this with a series of investigation in tractable murine systems, murine models of blood transfusion, in particular Dr. Jim Zimring at University of Virginia, Gary Churchill at Jackson Lab, and the Spitalnik and Hod Lab at Columbia University in New York. In particular, what did we do? First, we participated in this REDS RBComics study, which is an HLBI-sponsored investigation across 4 multiple blood centers in the United States, where 13,000 blood donors, ultimately 13,091 consenting blood donors, donated a unit of blood. This unit was stored for up to 42 days, and at the end of storage, this unit was tested for the hemolytic propensity, either spontaneous, just what percentage of red blood cells pops, hemolysis in the blood bag as a function of storage duration, or as a function of osmotic or oxidative tests, the classic pink test, or oxidant insult with the pro-oxidant AAPH compound at the end of storage. In particular, very relevant to this study is that not only we measured these hemolytic propensity parameters, but also we capture genomics data for 879,000 single nucleotide polymorphisms to afford the association of hemolytic propensity to genotypes. In parallel, our lab, through the very high throughput approaches that I described before, was asked to run metabolomics on all the 13,000 blood units. And here is a 3D map of the constellation of blood donor metabolic phenotypes, each single dot representing a blood unit or the combination of metabolic phenotype of any given blood unit at the end of the storage duration, storage day 42, which is a shelf life for blood units in most states in the United States. And as you can see here, you literally have a whole constellation of donors based on metabolic phenotypes. What is informing this distribution? First from last, I showed you that as storage progresses, red blood cell change their shape as a function of storage duration. So first, we set out to understand the extremes in this cohort. So we picked the donors with the lowest and highest hemolytic propensity. So those donors ranking below the 5th percentile and above the 95th percentile based on the 3 hemolysis parameters that I mentioned before. And we invited them to donate a second independent unit of blood, which was stored again for up to 42 days and tested, sampled sterilely at storage day 10, 23, and 42. So we have longitudinal sample from 643 whole donors for which we ran metabolomics, proteomics, and lipidomics. for the sake of today's presentation, I'm mostly going to be focusing on the metabolomic analysis. And storage progresses, what you see is that from top to bottom, from fresh blood to the end of storage, you have small molecules that go from blue to red, meaning metabolites that accumulate in the blood bag, and metabolites that go from red to blue, so small molecules that are consumed by the red blood cells as a function of storage duration. And you can actually identify metabolic markers of the so-called metabolic age of the blood unit. And based on these metabolic markers, you can literally write algorithms that can predict the storage state or the metabolic state in which the red blood cell is at the very moment in which the analysis is performed. We can actually use this combination of metabolic traits to prospectively validate the metabolic age of the units across multiple centers. And we did this across 10 multiple centers in multiple validation studies across different additive solutions, and ultimately use this information to understand whether a specific additive is impacting the rapidity to which the red blood cell metabolism age in a blood bag as a function of storage duration, and ultimately inform the development of novel storage solutions for improving the quality of blood products. Going to specific markers of the metabolic age of a blood unit, one that stood out in particular is hypoxanthine, which is a breakdown product of adenosine triphosphate, the main energy substrate for any and all reaction in mature red blood cells, along with GTP. But ATP is by large and far the energy token in a mature red blood cell. And ATP can be consumed into lower energy phosphate compounds like ADP and AMP, and ultimately AMP itself can be deaminated or break down into adenosine and then dominated into inosine, and then from there into hypoxanthine. So ultimately, hypoxanthine represent metabolic marker, both of energy starvation, energy depletion, and oxygen stress in a mature erythrocyte. You can actually use these predictive models to, again, validate prospectively the metabolic age of the unit, and it's been patented and is available there for determining the quality of newly issued blood product at scale. As we are planning to collaborate, for example, with creative testing solution to be using these parameters to test all the 15 million blood units that are donated in the United States every year. Why is any of this important, if it is important at all? In the second half of the presentation, I will try to convince you that it is translation relevant and why. First, I already mentioned that ATP, adenosine triphosphate, is pretty much the molecular energy currency that is used to virtually fuel any and all reactions in mature blood cells, from fueling ion pump to promoting proteasomal dependent degradation for removal of irreversibly oxidized protein components. Indeed, while it is true that our blood cells cannot synthesize de novo proteins, they do rely on the removal of oxidally damaged component as they progressively age, both in circulation or in a blood bag. And these processes are in part regulated by adenosine triphosphate. I mentioned the interactome with red blood cells response that varies in response to epoxy and oxygen stress, and that too is regulated by processes of ATP-fueled phosphorylation. I will spare you all the details of anything that is regulated by ATP, but if you're interested in reading more about this, there's plenty of reviews that I summarized at the bottom that myself, I wrote in the last few years. And obviously, many other colleagues have written something to this extent over the last few decades. What you may not be necessarily familiar with is that ATP in red blood cells derives exclusively from one single pathway, which is glycolysis, or an Erdem-Meyer-Parnasse pathway, in that red blood cells, unlike any other cells, lack mitochondria. We actually know that now that there are exceptions to this assumption in that, for example, in the context of sickle cell disease and systemic lupus erythematosus and other pathological conditions, we see up to 5 to 6 somewhat functional mitochondria in the mature erythrocyte. And it's a problem when you have mitochondria that consume oxygen as a final acceptor of electron in the very cell that is loaded with oxygen in the first place because of its very physiological function in transporting it to peripheral tissues. But that's a matter for another day and another presentation. However, what is relevant to this presentation is that glycolysis, as we break it down here in the rats RBComics in their index cohort of 13,091 blood donors is impacted by donor age, the x-axis here in each one of these graphs, and donor sex, with sex dimorphism showing, for example, impact on specific rate-limiting substrates, for example, glucose itself or lactate, with a sex dimorphism being evident at least until age 51, which is the average age for menopause in the United States. So I told you in the first few slides that there was an impact of storage duration and an impact of additive solution, the quality, or at least in the metabolic phenotypes, sort of blood units, but also from these large-scale studies, we can start appreciating the impact of donor demographics, such as donor age and sex, on the metabolic profiles in stored blood units. And actually, we can then use K-means clustering to, in an unsupervised fashion, leverage metabolomics data to identify what are the main subgroups that, based on metabolic phenotypes, identify subpopulation of blood donors. And for the sake of today's presentation, I'm going to be focusing on this subpopulation here. That is the most distinct one from the remainder of the rats RBComics index cohort. In particular, this cluster 4, arbitrary number that came out of the K-means clustering, seems to be characterized by a subgroup of donors with elevated adenosine triphosphate, or ATP. So a sort of group of super donors that have high ATP levels independently of sex and age of these donors. And high ATP was associated with an overrepresentation of donors with low hemolysis in this subgroup, as opposed to high hemolyzer not being represented in this subgroup. And not just hemolysis in the blood bag, but we can actually perform in an independent cohort, the donor iron deficiency study run by Dr. Eldad Hod at Columbia University in New York, whereby chromium 51 radiolabeling and metabolic characterization of the units at the end of donation, right before, at the end of storage, right before chromium 51 studies, identified metabolites in glycolysis, exophosphate isomers, and lactate, and ATP as the end product energy currency of this, as positive correlates to the higher post-transfusion recovery, the higher percentage of transfused red blood cells that still circulate 24 hours after transfusion, and vice versa, hypoxanthine, the oxidation and breakdown product of ATP, as the top negative correlate to post-transfusion recovery. I told you that these studies are performed in autologous healthy donors, so the donor is also the recipient of their own blood. What do any of these parameters and measurement and correlation associate to functional outcome in non-autologous, non-healthy recipients? To answer that question, we collaborated with Dr. Narek Grubinian, Kaiser Permanente UCSF, to look at the so-called vein-to-vein database, where we are linking metabolomics parameters, and I'll show you later on genomics data, to hemoglobin increments. So how much hemoglobin increases per unit of blood unit transfused after single unit transfusion in real-world transfusion recipients, so clinical patients requiring transfusion, including critically ill patients. And what we did find is that elevated ATP or low hypoxanthine is associated with higher hemoglobin increment, so higher efficacy of the blood unit transfusion, a lower bilirubin increment, lower post-transfusion in-circulation hemolysis, or extravascular hemolysis, for which bilirubin is an indirect marker, in the vein-to-vein database cohort. And interestingly, we can link ATP and hypoxanthine level to specific genetic traits that regulate them. I don't have the time to provide all the details about this today, but if you're interested, the paper was published last year in Cell Metabolism. And in particular, we found that a specific isoform, the phosphofructokinase, PFKP, which was originally thought to be a contaminant from platelets in mature blood cells, but now that we have this ultra-pure red blood cell population, we know that it's indeed present at approximately 11% in mature blood cells, and donors carrying two alleles of the PFKP specific of a specific PFKP SNP single nucleotide polymorphism actually have higher quality of their donated blood, which result in higher hemoglobin increments after transfusion. You may argue that, to be able to account for all the factors that confound the interpretation of the quality of single product transfusion in the real world, you would need millions of transfusion events. And I would agree with you. So how do you go about mechanistically testing the role of specific parameters, for example, genetic traits beyond PFKP, in a controlled setting without confounding factors such as, for example, donor age, sex, BMI, or exposures, which we'll get to in the last part of the presentation. One way to go about that is use tractable murine system. We did so with a model we optimized with Dr. Jim Zimri, University of Virginia, whereby you test blood from a donor mouse strain. You store their blood for 7 to 12 days, equivalent to day 21, day 42 for human red blood cells. And then you transfuse these red blood cells into UBGFP mice, where all the red blood cells are green fluorescent labeled. And we tracer a cell, fresh red blood cells from UB cherry mice. So all the red blood cells being red labeled. You can do an alternative to that where the tracer population is actually biotinylated instead of cherry labeled. There are some caveats to that, but we'll go around these details today. And ultimately, you can use all of this to look at the heterogeneity in storage quality and post-transfusion performances, post-transfusion recovery, that mimics the chromosomal 51 labeling studies in humans by interrogating the impact of genetic traits across mouse strains with the most extreme genetic diversity. We started with 13 different mouse strains and identify ultimately some mouse strains that store really well and some mouse strains that store really poorly. What you can do next is a series of things. First, you can identify the mouse strains, including diversity of bred mice, in collaboration with Jackson Lab, in particular Dr. Gary Churchill, identify the most extreme of these founder strains and cross this collaborative cross 8 founder strains up to, we did 35 generation, and then in a follow-up study up to 46 and counting, to obtain 350 mice, diversity of bred mice. There are genotype of 137,000 polymorphisms, and for which we run metabolomics, lipidomics, and proteomics. We'll not have a chance to talk about the proteomics pQTL analysis today, but what you can do is you can start linking metabolic and lipid traits to genetic parameters and ultimately identify specific genetic hotspots, including one in particular that maps on chromosome 1 in stored red blood cells. We did this both in fresh and stored red blood cells, that ultimately maps on a region coding for this ferry reductase, STIP3, coded by gene on chromosome 1. Interestingly, the post-transfusion recovery had the strongest negative correlation with lipid peroxidation products, and all mapping, like I mentioned before, on this region of chromosome 1 coding for the ferry reductase STIP3. And it just happens that we found multiple haplotypes for STIP3. One in particular is the single amino acid substitution across mouse strain for alanine 350, in alanine 350 for a valine, which impacts the activity of the ferry reductase as measured by ferrozyme reduction assay. An additional thing you can do from a genetic standpoint is that once you identify the good story mice and the poor story mice, you can cross them directly, and then the F1 population that you obtain, you get the poor story mice and cross them with the good story mice again, and go to an F2, F3, F4. And this actually project took Jim Zimring's lab in particular over 5 years, and we were running metabolomics as we went, and ultimately identified that a region on chromosome 1 of 4 kilobases in a region coding once again for STIP3, and we could actually CRISPR in the specific gain of function allele for STIP3 into the good story mice and recapitulate the poor storage quality of the poor story mice or the F1 population obtained from the crossing of good storing and poor storing mice. And all of this resulted in not just poor post-transfusion recovery, but excess lipid peroxidation. So now we start accumulating evidence that an iron-dependent enzyme is linked to lipid peroxidation and linked to increased extravascular hemolysis of red blood cells because of excess lipid peroxidation in this population. You may argue, well, this is relevant in mice, but is it relevant at all in humans? So we went back to our rats RBComics index cohort and we looked for metabolic predictors of hemolysis. And lo and behold, heats and oats, hydroxyoctadecadenoic acid and hydroxyeicosapentaenoic acid. So lipid alcohol derived from oxidation and detoxification of lipid hydroperoxide of the 18,2 and 24 fatty acyl chain were the strongest predictors of red blood cell hemolytic propensity in the bag or after oxidant stress. And this hemolytic propensity was associated with over 27 single nucleotide polymorphism for STIP3, including specific missense mutation on the region coding for STIP3. Using heats and oats, so lipid peroxidation product and lipid alcohol derived from it, as quantitative trait for association of metabolite as QTL, so quantitative trait loci analysis, in a GWAS-like approach, we can identify the genetic traits that are associated with heterogeneity and higher or lower lipid peroxidation. And lo and behold, while STIP3 did not directly come out from this analysis, mostly because some of these traits, if in homozygosity, preclude you from donating blood in the first place. We did observe that a series of enzymes in well-established ferroptosis cascades are involved, are coming out from this analysis. For example, GXPD, glucose-6-phosphate dehydrogenase, which generates reducing equivalent NADPH to reduce oxidized glutathione, but also to fuel STIP3, which is an NADPH-dependent ferric reductase that converts ferric to ferrous iron. So it's kind of a double-edged sword there. We also identify glutathione synthetase or carnitine transporter like SLC2216 or lysophosphadylcholine acetyltransferase that uses very long-chain acyl-CoAs to fix the lysophospholipid damage upon generation of lipid hydroperoxide that are chopped off from the membrane through phospholipase A2 activity or phospholipase A2-like enzymes. In particular, we also found that very long-chain polyunsaturated fatty acids are associated with a higher degree of unsaturation, and the enzymes that participate in the elongation or the desaturation, enzymes that are present and active in our deep proteome analysis, are also associated with the degree to which lipid peroxidation accumulates in stored blood units. And ultimately, the donors with the highest levels of heats and oats had the lowest hemoglobin increments, especially if those donors, if those units were stored longer than 3 weeks and came from donors carrying specific genetic polymorphism in LPCAT3 or other enzymes in this cascade. So from a translational standpoint, you can actually use specific labeling approaches like CFD-ASC to get a mixture of red blood cells at the end of storage, and the ones that cannot use active ATP to pump out specific fluorophores, CFDA that becomes CFSC once it forms adducts, can be flow sorted out and separated from the remainder of the population. And ultimately, you can look at the fresh red blood cells or the stored red blood cells that are morphologically preserved versus the ones that are morphologically altered and fluorescent positive. And everything that I told you goes bad with storage goes bad specifically in that subpopulation. And the size of the subpopulation is going to impact the degree to which you have lipid peroxidation and you consume acylcarnitines to repair the lipid peroxidation damage through the so-called Lent cycle in mature erythrocyte, and the degree to which the stored red blood cells, so how large is the subpopulation of stored red blood cells that get sequestered in the spleen upon transfusion. We did this both with chromium 51 radiolabeling and with ex vivo perfusion of spleens from organ donors. in collaboration with Dr. Pascal Amiro and Pierre Buffet at Institut Pasteur and INSERM in Paris. All these observations are not relevant just for red blood cell storage biology, but also for red blood cell aging in circulation, to the extent that the identical phenotype of increased lipid peroxidation and depletion of carnitine pools and alteration of the Lens cycle pathway are observed in murine and human red blood cells as they age in circulation, as determined by biotinylation studies or Percol density gradient studies. You can use all this information both at the metabolome, lipidome level to in silico simulate the membrane deformability and then in so doing, using omics-based tools to predict whether a red blood cell is gonna theoretically be deformable enough to squeeze through the splenic slit upon transfusion. And since you can simulate all of this in silico, you can actually come up with strategies to boost metabolism in a way that fixes the damage to some extent, at least the damage that is still reversible. And for example, contribute to the repairing of lipid peroxidation product through the acylcarnitine pools by introducing L-carnitine in additive solutions, which doubles the post-transfusion recovery. We see this, of course, with L-carnitine, but not with the non-active enantiomer D-carnitine. So by now, I should have convinced you that a lot of things changes with storage and are relevant to outcomes, but is that really the case? When we look at the simple association of metabolic traits to hemolytic propensities, either spontaneous, osmotic, and oxidative, we see that the metabolites that change the most with storage are not necessarily strongly associated with the hemolytic propensity, maybe with the exception of the aforementioned hypoxanthine. So what does associate with hemolytic propensity? One explanation for that is that even though the trends over storage duration for all donors are consistent, the slope, onset, progression, and ultimate severity of the metabolic storage lesion varies from donor to donor. In addition to all of that, when we associate parameters like osmotic fragility to metabolic phenotypes, we see that metabolites like kynurenine are strongly associated with hemolytic propensity, in particular osmotic fragility, despite kynurenine not changing at all as a function of storage duration. For those of you who are not familiar with kynurenine, it is a tryptophan metabolite that is metabolized, synthesized by indolamine 2,3-dioxygenase or tryptophan dioxygenase. And in our analysis, in the RADS RBComics cohort, is one of the top markers of red blood cell donor age and the top marker of blood cell BMI. So the highest BMI donors have the highest kynurenine in the first place. And it's also showing a sex dimorphism at least until the age of 51, once again, the average age for menopause in the United States. If you're interested in reading more about metabolic markers of aging and circulation, we can actually use all this information from Metabolomics data of the 13,000 blood donors and multiple cohorts on top of this RADS RBComics study to identify a so-called aging clock that can predict the circulating metabolic age in humans. Going back to our kynurenine, one particularly interesting finding is that when we look at the reproducibility of metabolites across multiple donation, acylcarnitines, which I just mentioned in the context of lipid peroxidation, and kynurenine are amongst the top metabolites, actually top 10 metabolites that are most reproducible within across multiple donation. There are 6-12 months apart from the same donor. Which suggests there is a strong genetic component that is donor-specific that impacts the levels of kynurenine as a metabolite in a blood unit. And indeed, when we map kynurenine levels in 13,000 donors on genotypes, we find that the tryptophan or kynurenine itself transporter, LAT1 or SLC7A5, but also the rate-limiting enzyme for kynurenine synthesis or catabolism, ADO1, TDO2, KMO, kynurenine monoxygenase, are all identified as genetic traits that are polymorphic in blood donors and impact kynurenine levels and ultimately post-transfusion recovery and osmotic fragility. We can repeat this analysis that I just showed you, QTL analysis or metabolite quantitative trait loss analysis, MQTL, not just for kynurenine, but also for all the other metabolites in stored red blood cells from the 13,000 donors. And this paper is in preparation, will come out in the next few months, Well, ultimately, we do find that the metabolic heterogeneity of blood units is strongly genetically encoded with over 1 million gene metabolite association across blood donors. And we're using all this association to rewrite literally the textbooks of red blood cell metabolism. Very interesting from a translational standpoint, we see that blood donors are actually often carriers of, at least heterozygote carriers, of alleles, including missense mutation for specific traits that are otherwise associated with inborn errors of metabolism. And whether they are healthy enough to donate blood, so they have no patent manifest metabolic defects, abnormalities, storage is a stress sufficiently harsh to unmask the metabolic defect that is otherwise present, even in donors carrying these traits in heterozygosity. For example, here I'm adding the branched chain ketoacid dehydrogenase, which is the etiological contributor to maple syrup urinary disease. Of all these, Mendelian traits, the most interesting one and most prevalent one we found is kind of obvious. Again, glucose 6-phosphate dehydrogenase, the rate-limiting enzyme of the pentosphosphate pathway, in particular here strongly associated with osmotic fragility and oxidative hemolysis. A particular prevalent in donors, at least in this cohort of African descent, and in other cohorts in Europe in collaboration with Italian and Greek colleagues in the Mediterranean region. Indeed, the Mediterranean variant serine 188 phenylalanine. In conclusion to this part of the presentation, we believe that donors are carriers of traits that are otherwise associated, that are otherwise silent through their lifespan, but can be unmasked by stress. Stress can be, in this case, in this presentation, storage in the blood bank, but this stress can be manifesting at any time in the donor's life. For example, if they go and run an ultramarathon, or if they change their diet, or just by simple inflammatory mechanisms in aging, these donors can start developing conditions that are otherwise more prevalent in people carrying both alleles in homozygosity of the specific etiological traits etiologically associated with the specific inborn error of metabolism. And ultimately, all of us, as we age, we develop conditions that seem to come out of nowhere, but likely are explained, at least in part, by this phenomena at the gene metabolome interface. At least in part, I mentioned, for example, STIP3. The other name that you may know STIP3 for under is T sub 6, where T is tumor protein. So TP53, tumor protein P53 associated secretory antigen protein. So looking at the genetic traits that were associated with metabolic abnormalities, in particular lipid peroxidation, we found that beyond STIP3, P53 mutations, including some missense mutations that are predisposing to cancer, were prevalent in the blood donor population with similar prevalence as it is observed in individuals who eventually develop cancers. So now we are seeing information, we're gathering information about donor biology that can tell us that at some point, provided specific prophylaxis is not implemented, donors may stop donating blood at some point because of, for example, development of specific metabolic disorders, or in this very case, with P53 being in linkage disequilibrium with all the ferroptosis associated traits, excess lipid peroxidation, or even predisposition to develop cancer. And we find that these traits, all of them, from G6PD to STIP3 to GPX4 to P53, all the missense mutation and all the factors that were predisposing to lipid peroxidation and ferroptosis and increased hemolysis were all predominant in donors of African descent, both from genetic ancestry and self-reported ethnicity. I'll close here with the next two slides. We believe there is more to this story. We believe there is more than just the genetics and demographics of the donors. In particular, when we combine the metabolomics data on kynurenine, the top predictor of osmotic fragility, with the proteomics data, we see a strong positive association with protein components linked to inflammation and viral responses. Indeed, kynurenine level were not only the top markers of osmotic fragility, but also the top predictor of donor obesity and aging. And in parallel studies that we conducted over the years, we find that kynurenine levels are strongly regulated by interferon responses, which is the direct transcriptional regulator of IDO1. And we see that in conditions whereby type 1 interferon responses are upregulated, be it inflammation, be it aging, be it trisomy 21, 4 out of 6 of the interferon receptors are coded by genes on chromosome 21, or even like infections, either with coronaviruses, including SARS-CoV-2, or flaviviruses, including Zika, which is potentially transfusion transmissible, are all associated with elevated levels of kynurenine in the blood unit, even months after the infection has resolved. So in going back to our map of the red blood cell metabolome and the K-means clustering, I showed you that there are groups of super donors, but also a series of other clusters that we haven't identified. And when we focus on those clusters, we see that those clusters are rich in donors who smoke, drink alcohol, drink coffee, and we find that these metabolites of exposure are impacting the red blood cell metabolome in a way that influences, for example, the red blood cell capacity to cope with oxidant stress, for example, by activating or inhibiting the pentose phosphate pathway. Beyond recreational professional habits, professional exposure or recreational habits exposure, we see that medical iatrogenic exposures, drugs that are not ground for blood donor deferral, are routinely detected in blood units at non-zero frequency. For example, we see blood pressure medication, we see anti-inflammatory drugs, acetaminophen, ibuprofen, we see statins, we see antacids, all extremely common in blood donors at different age and demographics. Ultimately, what any of these residual drugs do, whether the drugs themselves or their metabolites or the impact they have on the metabolic age of the blood unit, whether any of this is relevant to transfusion biology with respect to safety or transfusion outcomes, still matter of debate. And hopefully this whole field, the exposome that we started in collaboration with others in the last few years, will offer some answers in the upcoming months to years with adequate clinical trials testing specific hypotheses. So in conclusion, all we have learned is that things change with storage, and we understand now specifically what changes with storage, and how we can cope with it by preventing the storage lesion in the first place, or restoring the storage lesion to the extent that the storage lesion is reversible through rejuvenation strategies to make erythrocytes great again. In conclusion, we believe that all these studies combined offer a new appreciation of the complexity of red blood cells beyond red blood cells as a simple cell, and red blood cell as a reappreciated marker of systems metabolism, in keeping with the concept of chemical individuality that was foundational to the whole field of clinical chemistry and inborn error on metabolism. We believe that we can leverage combined information both at the genome and metabolome level to first improve inventory management, for example, in a data-driven fashion. This blood unit is better stored in this additive solution, is better stored for 3 weeks rather than 6. This unit can be stored for longer because this comes from super donors. These additives is going to improve specifically blood coming from the donors, but not others. Also, this information is going to offer an opportunity to provide information back to the donors to keep them in health, not just selfishly to make them keep donating blood over time, but also, I think first and foremost, to give something back to these altruistic volunteers who have been saving lives with their gift of blood donation. Beyond transfusion medicine, we believe that leveraging blood donor as a generally healthy population, the center of this Gaussian curve, and integration of data, metabolomics data at scale from unhealthy patients in any of the hospitals at CU Anschutz on campus, on one side of the spectrum. On the other side of the spectrum, athletes from recreational to elite athletes, the most perfect machines, Formula One cars amongst all the cars out there, if you believe the analogy of metabolism as a very efficient car. Understanding this continuum of health or this continuum of metabolic health can offer insight on how to improve metabolism at scale with potential benefits beyond transfusion medicine. Ultimately, concluding that information about blood may soon be almost as important as blood itself. With that, I'll be happy to take any question, and thank you for your attention.
Wonderful. Thank you so much.
much.
Go ahead, Anna.
Oh, I was just going to say questions for Dr. D'Alessandro.
Go ahead, Jonathan.
Just a quick question. Thank you so much. The tryptophan metabolite that accumulates is part of the NADH pathway, I think. There's a precursor if I'm... And I think what people have done in the kidney AKI is they've tried to resuscitate by giving niacinamide treatment. I don't know if that has any effect on red cells.
Well, we haven't tested it yet, so I don't necessarily know the answer to the question. However, de novo synthesis, NAD synthesis in red blood cells is incomplete. So, we tried using my riboside, another precursor, to push more on the salvage reaction. Others have tried to push more on the price handler, but worse in our hands, at least, for example, in the context of leukemia stem cell metabolism, where paradoxically we want to do the opposite, we want to target that, but we haven't really tried it in the context of red blood cell storage biology. When we use labeled tryptophan in incubator red blood cells with 13C15N labeled tryptophan, not much happens to it. Very little gets metabolized further. So the evidence we have is that that canurine that doesn't change during storage in the blood bank is in perfect correlation, and it's part of the paper, with the circulating levels of canurine. So we believe that canurine is just a manifestation of circulating levels of inflammation. The other argument you were making about canurine and NAD synthesis connection there, the association between canurine and aging is also important in the context of NAD, whereby NAD depletion is one of the hallmarks of aging, right? So we've been doing a lot of aging-related works where we've been, from parabiosis in mice all the way to intervention in aging mice to specific manipulation of genetic traits, including G6PD status in the context of aging. We've been seeing an effect of manipulation of the pathway for sure.
We have a question, a raised hand from Russell Ware. Go ahead, Russell.
Angelo, such excellent, amazing work. I had two questions that are sort of related. The first is at the very beginning, you exclude all white cells, platelets, of course, but you exclude reticulocytes, all CD71 positives. Have you ever done this analysis just on the reticulocytes? Of course, they are the most metabolically active, but they're also most energetically dependent upon metabolism. And so you expose things, obviously, like in PK deficiency. So that was one question. And then related, have you thought about looking just at deficient erythrocytes in every step of the enzymatic pathway to see if there are new connections that maybe aren't as obvious from the straight linear pathways? Thank you.
Yes and yes. So I didn't show it today just in the interest of time, and apologize for going 5 minutes over, but we have similar deep red proteomics data exclusively on the reticulocyte fraction. And we are, of course, we are 2,000 proteins above what we see in red blood cells, right? At least. We're probably not going to include it. And the maturity red blood cell fraction is different between healthy individuals versus sickle cell patients, right? Patients with sickle cell disease, I apologize. So, and we are doing now repeating in copy paste and all sorts of conditions from chorea acanthocytosis. We published literally this morning on maple syrup urinary disease. but propionic acidemia, pyruvate kinase deficiency, you name it. And actually, I'm welcoming, if you have any specific case you're interested in, we're going to look at the red blood cells. We would love to. Please send over. We can literally throw everything but the kitchen sink, and then use these models, these proteomics data in particular. But we are now moving from proteomics to metabolomics. So we're generating in silico models that are not genomically informed, but informed by the actual proteomics data. And we're using the estimated protein copy number per cell to inform the red blood cell gem, so a reconstruction of an in silico red blood cell, and using that to inform how the metabolic fluxes are rewired to come up with specific intervention as a function of the specific metabolic abnormality. Unfortunately, that is limited by several limitations. For example, it does not account for specific polymorphisms and that missense mutation that impact, for example, the activity on enzyme and the variation of KM for specific enzyme as a function of this. So the next generation of RBC Gem 2.0 is actually going to include that, but it's 3-4 years in the making. And in addition to that, we are trying to obviate all of this by using metabolomics data, in particular metabolic flux data, to probe the fluxes through specific pathways as a function of these traits. So we can now inform how specific enzyme kinetics are impacted by specific alterations of genes. The first generation of this model was just eliminating in silico one enzyme, but that's not how nature works. Because, for example, in GXPD deficiency, you compensate by upregulating an NF2 dependent target. So now you have PGD upregulation. You have anyway. You will have to shut me up. Sorry, I get excited and I keep talking.
Clearly, these conversations should continue. This is fun. Any other questions? I don't see anything in the chat, so I think we can call it good. And Anna, I'll let you close us out.
Yes, I think that's it that's all the time we have for today. But thank you so much, Angelo. That was awesome. We really appreciate your time. That was a really informative and fantastic seminar. Thank you so much for joining us.
Just remind everybody that this recording will be posted on the CIHD's YouTube channel, which you can go look up when you get a chance. And so nice to hear the stories. Really thought-provoking. Really, really thought-provoking.
Really comprehensive work. It was really fantastic. Yeah. Thank you so much, Angelo. Really appreciate it.
Have a good day. Bye-bye.
Have a great day. Bye-bye.